Roma ma'am, you on to you, ma'am. Now over to you, and you welcome Mirza sir and read her profile.
Good evening, everyone. So I'll just introduce you all to the
voice clear.
speaker
Okay.
of session
For
3.
inviting me.
Session 3 speaker, Dr. Mirza Sarwar Beg. So sir is basically working in the department of Jamia Millia Islamia, New Delhi, in the field of uh, 3 departments: molecular biology, virology, and applied bioinformatics. And he is basically skilled and in various molecular biology techniques like reverse transcriptase PCR, real-time PCR, southern hybridization, dot plot, northern blot. And he is well versed in Perl scripts, R language for bioinformatics. He is aware about different softwares related to bioinformatics like Autodoc, Bina, Pyrex, Schrodinger, Glide, Samson, and he is into molecular dynamics simulation through Gromix. So sir had completed his master's and from in the field of bioinformatics from Hamdard University. Then he went for PhD from Department of Biosciences, Jamia Millia Islamia, on engineering the CLCD President's cotton plant manipulating silencing suppressor gene. He had published various research articles in national and international journals, and he had gone for various oral poster and abstract presentations in different national and international conferences. He had been the member of the academic counselor by the IGNOU, and he was given the Indian Council of Medical Research Associate Fellowship as well. He was given different other recognitions and fellowships by various different reputed organizations. So thank you so much, Dr. Mirza, for accepting the invitation. And the participants, today he will be talking on stepwise protocol to perform protein modeling, molecular batch docking, and virtual screening of drug likeness of small organic compounds. So I request Dr. Mirza to please start your session.
Yeah. Thank you, ma'am. Yes. My voice is. Yeah.
Yes, your voice is clear, sir.
Thank you for inviting me. Just I'm showing my presentation. Today's talk of my lecture is a stepwise protocol to perform protein molecular docking, batch docking, and virtual screening of a small compounds for drug likeness evaluation. Okay, so I'm going to present a stepwise protocol which I always use, which I use to do my computational molecular modeling and docking in my day-to-day computation. So I will try to convey the whole message of what is the basic philosophy of molecular modeling and why docking is so important and what is the significance of protein and their counterpart ligands. So share screen. Just I'm sharing my screen. Yeah. Is my screen coming? My screening visible on your computer screen? My slides are visible on your computer screen?
Yes, sir, they are visible.
Yeah, okay. Your voice is very slow, perhaps. So that's why I am not. Okay, now let's start. Now let us start from the molecular modeling. Why molecular modeling is going to be performed? There are two reasons of molecular modeling. If the protein of your interest or your protein, target protein is having X-ray crystallographic structure or NMR structure already available in the PDB, then there is no need of molecular modeling. We directly download the PDB ID, directly PDB ID from the protein data bank, and then we identify the missing residue, then we identify the protein active sites, etc., from the already known well-characterized protein. But most of the time, if you have a novel protein and the structure of your protein is not available in the protein data bank, then what you have to do, you have to first have model your protein of your interest, which is only in the form you have only sequence. So you have to check your sequence in the FASTA format, and then you have to go for any software or any online resource where you can put your protein amino acid sequence to get the exact scaffold of the protein with the most similar or most homologous protein. So in this context, there are different methods of molecular modeling. Here, homology modeling or comparative modeling is one of the most basic and one of the most important molecular modeling method. Another are known as threading or fold recognition, and another is ab initio prediction, and the last one is the transmembrane helix and peptide structure prediction. Because homology modeling is very famous and all softwares uh, which are working on online mode have adopted this method. Because if the protein of your sequence is having more than 30% similarity with the database sequence, then you are in a safe zone and you can go for you can easily go for molecular comparative modeling. But if your sequence of interest is having not more than 30% sequence identity with the database sequences, then there is a problem and you have to there is unsafe to go for the comparative modeling, and you have to use either threading or ab initio prediction. Here, in the case of threading or fold recognition, the whole sequence which you have entered is broken into several small scaffolds, and from the scaffold library, these small patches of amino acid sequences are then combined and assembled by the algorithm to get a model of your protein. So this is a very time consuming and sometimes this is error prone step, but it works for your own purpose of molecular docking. The last one is the transmembrane helix or peptide structure prediction. It is one of the toughest area of the protein modeling because the transmembrane proteins are very tough to be modeled, but still there are some algorithms available. By using these algorithms or some sophisticated algorithm, you can draw the structure of transmembrane as well as the peptide. So here some algorithms for protein structure prediction are known as Cho and Fassman algorithm, which is one of the most pioneer. Cho and Fassman are one of the most pioneer in the field of protein modeling. Because modeling of any amino acid nucleotide or DNA or RNA is simple because of only less variation in the less variants, only 4 amino acid, 4 nucleotides or 4 bases are available. But here in the case of protein, which is very dynamic entity, it has about 20 and more than 20 amino acid residues. So their permutation and combination of forming a exact structure in a very dynamical way is a very tough to model. But still, Cho and Fassman have written an algorithm based on the physicochemical properties of each and every individual amino acid and developed an algorithm to model the protein. But still, with reference to the DNA and RNA modeling and protein modeling, we can easily have a superiority in the modeling of DNA and RNA, and the accuracy of the softwares are more than 90%. But in the case of protein modeling, the accuracy is not so high, and it remains about 60 or 70% or not more than this. So still using this accuracy standard, we can move forward for our own, uh, for solving the problem of my own interest. So Cho, Paswan, Lim and Robson's and Cohen algorithms are known as the first generation of algorithms, and they are somehow less sensitive, while the King and Steinberg and Magellan algorithm, which was developed in 1990s, they are more sensitive, and they are known as the second generation of protein structure prediction algorithms. So now we will discuss what are the steps of homology modeling or comparative modeling, which is our own interest to do automated modeling. So homology modeling is comprised of 7 steps. Here, template recognition, it means the sequence upon which your sequence is going to be modeled is known as template, and your sequence is known as target. The query sequence will be known as the target, and the sequence available in the database with highest similarity is known as template. So template recognition is one of the initial step. Then alignment, then we have to use align these sequence, your own sequence, query sequence, using some algorithms, for example, BLAST algorithm, for example, HH hit BLAST, Psi BLAST, Psi iterative BLAST. So there are different type of basic local alignment search tool. Using this tool, you can align your sequence of interest with the database sequence. Then determining a structurally conserved region. This is very important to start your scaffold folding. The highest conserved region is modeled first, and then we extend from the less conserved region. So as identification of a structurally conserved region is another part. Then backbone generation is another process. When the conserved region is going to be identified, the seed of modeling starts, then of whole and soul backbone of amino acid peptide backbone based on phi and psi angles, which is known as dihedral angles of any peptide molecule starts. So backbone of your protein is going to be generated. Then the side chains known as loops or variable regions are modeled. Then side chains of your protein of interest is going to be modeled, and their conformational stages are going to be decided. Then in the 6th stage, your structure is going to be refined on some certain parameters. So refinement of the structure is also whether your some amino acid residues are missing or having replacement of some amino acid or other there is steric clashes or other structural or so a structure validation and refinement of these two are important steps of the homology modeling. So we will proceed according to these steps of homology modeling. There are several automated web-based homology modeling servers which we can prefer, in which sys model workspace is one of the most cited tool now these days, which is going to be used for easy molecular modeling with high accuracy and precision. So sys model workspace is based on the template detection, alignment, and 3D modeling by its own. So Swiss model workspace, which is the copyright software of XPASI, it is one of the best modeling tool. Previously, Modeler was very famous in molecular modeling, but still Modeler needs much more intervention of the user, but Swiss model doesn't need much more intervention of a user. Rather, it computationally it is so robust that in a single click, by single click and click mode, you can do the modeling of your protein very easily. So there are lots of modeling servers by using which we can model our protein. So I will prefer, or most of the time I usually prefer to model of my protein of interest using sys model workspace. It is a very automated and very user-friendly user-friendly server from which you can model your protein. In the first window, you have to put your sequence in the FASTA format. So just we have kept our amino acid sequence in the FASTA format, and we have because here in the FASTA format, we don't have any heteroatom or ligand or a water molecule or etc. So here only 20 amino acid characters are going to be written in the FASTA file format. Then we have to search for the templates. After clicking the search for the templates, there are the execution of template searching starts. Here you can easily see that there are different searching parameters on which this query sequence is going to be searched with the whole database sequence available in the protein data bank till the date. It means the most updated and revised protein data bank, which is highly organized a structural database. It compares your sequence from the protein data bank sequences and identifies the highest homologous or highest similar sequence from the database. So in the next step, what we have to do, we just this Swiss model workspace identifies that your sequence is having 32 templates. For example, I am showing here that there is 32 templates. It may be more or less. So when you put your protein of your own interest, it will give you the list of the total templates and some parameters. For example, GMQE, QSME, identity, method, oligostatus, and ligand. On these 4 or 5 parameters, you have to judge by your own, or the software identifies that most of the time global mean quality GMQE is one of the most and best parameter on which the protein is going to be identified. So highest GMQE score and identity is one of the safe parameter. By clicking or by checking this checkbox, you can identify that the topmost template is one of the best and safe template to build your model. Again, when you can use more than one template as well, there is no problem. More than one problem is also valid, but after selecting so many templates, there are large number of models will be generated and having some differences. So it will be trivial or very tough to identify the best possible model. So we prefer only a single template for modeling of the protein. Then after clicking the build model, the protein model is going to be generated. Here in this case, after 32 selection of 32 templates, only one model is going to be built. But if you select multiple templates, then it will build multiple models. So it will become trivial and confusing. So here you have to identify that the GMQE value and QME value are good enough, and the sequence similarity with coverage is very high. Then you can easily go for safe modeling. In the next step, if this model is going to be built on the same server, you have to validate your protein model. Here the structure assessment is going to be done by your own. By single click, you have to select the model and then go for the structure assessment. Here for a structural validation, Ramachandran plot, our 2-dimensional plot, is very robust for getting the percentage of amino acid residues, which is 2 dihedral angles phi and psi. By depicting these 2 phi and psi and the area of within the phi and psi 4 sectors, you can identify that the most allowed region is dark green and less light green is less allowed region, and exceptionally allowed region is white. So if the most of the amino acid residues is coming in the green safe zone, or more than 90% residues are coming in the Ramachandran most allowed region, then this modeled protein is is very accurate to be used for the docking purpose. So here in this case, Ramachandran favored region shows that there is 95.92% amino acid residues are coming in the favored region or allowed region. So this model is very good, but still there are some outliers and rotamer outliers and bad bonds, bad angles, etc. No problem. We can rectify these bad bonds and bad angles in other software by applying Autodoc or by applying other online tools and server. We can modify or we can rectify the missing elements or the missing amino acid or the bad angles and bad bonds using Swiss PDB viewer, by energy minimization, and there are several different softwares on which we have to now work on this model for its proper energy stability and energy. So here, upon analysis of all these criteria, we are now in the state that the protein is now, our model is enough good, and we can proceed for this protein for our docking purpose. Okay, so other model evaluation for evaluation of your protein model, there are other different online servers are available in which you can put your model of the protein and you can analyze whether how much differences and how much error are present in your model. So these different model evaluations servers are available, and you can use by single click, go and click. Now, the most important part of our talk is what is docking and why docking is important. Docking is a natural process in which most of the time people know that enzyme and their ligands most of the time are all the enzymatic activity of any protein and its ligand depends upon their binding affinity. So there may be different type of, I'm not going to discuss the biochemistry of the enzyme and ligand, enzyme and its ligand biochemistry. Rather, I'm just going to mimic this biochemical process on a computer. So mimicking a biochemical process in a computer is still not very satisfactory, but still there is a chance to mimic the process and we can infer some idea for our computational work. Here, docking is important for better understanding of for the machinery of life, enzyme, inhibitor classes, antibody, antigen classes, and other different type of interactions. So protein ligand interactions is very helpful in engineering protein enzymes and protein therapies and drug target identification and drug designing and drug discovery in large search space. If there are millions and billions of small organic compounds, then we can, how we can correlate and how we can sort out which compound or which organic compound is more feasible to inhibit or to activate. Both are possible. So inhibitors, which most of the time in the docking process, we use ligand, we term ligand as an inhibitor. So in the millions of compound for our
Excuse
own.
me, sir.
Yeah, sorry.
Can you just please expand this PPT, make it large, so that the audience wants the content is not visible to them.
Okay. I've shared the PPT.
PPT is shared. Just say आप बड़ा कर लीजिए F5 करके.
Okay. Font size.
No, make it visible. The only the page of the PPT should be visible. ये जो
The problem is not visible.
You're using your laptop or mobile?
No, I am using on a laptop.
Okay, sir, there is one icon here on the magnifying thing ना at the bottom of this माइनस साइन के लेफ्ट में, जस्ट उसे क्लिक कर दीजिए तो एक्सपैंड हो जाएगा स्क्रीन, पीपीटी फुल स्क्रीन हो जाएगा. Yes, yes, yes.
Thank you. Sorry. Okay. So engineered protein, enzyme, protein therapies, and so on, blah and blah. So on using a computer, we computational modeling, we can mimic the original biological phenomenon or biochemical phenomenon, or in a limited or using a very limited resources. So if you can use individual enzyme and ligand biochemistry through experiment, it is a vast, expensive work to identify any proper ligand for any proper inhibitor. But a computational biology gives you freedom to sort or virtually analyze millions of compounds for a specific protein, and then select a sort out top 1, top 10 molecule for your own experimental purposes. Okay. So previously the drug discovery was very trivial and it was based on the hit and trial method. But nowadays, due to the computational molecular modeling and simulations or other expensive computational analysis, the drug discovery and rational drug design is now very logical, and it is less time consuming and less resource consuming. So effort to cut down the research time and cost by reducing wet lab experiment, we prefer molecular docking. Okay, so the traditional drug designing was very trivial. Here, lead molecules are going to be generated. They may be the natural ligand, and they are going to be screened by wet lab experiment or biological testing on animals, model, by different various means, by oral or by other way. So orally, we have to feed the animal, and then you have to analyze the different physiological changes in the animal, and then we decide that okay, this medicine is harmful or this drug is harmful and it is having side effect, and this drug is beneficial, it is going to be improved. So the drug designing cycle was time consuming and very resource consuming. And if after all, when using this process, if you identify any compound, then you may, if it is found promising, then you go to the preclinical stages, etc. But now the structure-based virtual screening or rational drug design, here we can identify large number of natural or organic molecules available in PubChem, Zinc, or a large number of several databases where are millions of molecules. We can cluster or we can use these molecules using a Perl script or a Python script. We can fetch the database locally on our own Autodoc machine, and we can, if machine with high configuration, and then we can do our batch docking throughout the whole database. So especially here, you have to screen the total number of organic compound available in different public libraries, molecular libraries, which are almost free of cost without any investment of any penny. So here a special emphasis is given on the molecular biology of the protein and chemistry of the organic molecules. This is also a very important part. We cannot just randomly identify any biological protein or molecular, but rather we have to select a protein based on certain facts or certain rationales why we are selecting this protein, what is the function of this protein, what is the biochemistry of this protein, and all this molecular biology of protein should be extensively studied through knowledge bases. So we have to, for selecting any protein, we have to become very rational, and then chemistry of each and every organic molecule should be considered very robustly. Then 3D structure of the target protein is going to be modeled. If the protein is available in the database, PDB, or any other protein structure database or macromolecular database, then there is no problem. But if a structure of any protein of your interest is still not going to be determined by NMR or X-ray or X-ray crystallography, then we have to prefer the homology modeling or molecular modeling. Then the 3D determined 3D structure is now going to be docked with the ligand. then the first part of this rational drug designing is docking, and another part is molecular dynamics simulation. Not on only relying on docking is not sufficient. Rather, we have to simulate this protein ligand complex for different time frame in a different type of force field. Still, docking is going to be performed in a very it is not so much dynamic phenomenon, while molecular dynamic simulation is highly dynamic phenomenon, and it mimics the natural phenomenon very well. So docking is still very having some compliances, rigidity issues, and resource and computation power issues, while MD simulation uses high computation power and is more reliable for getting the protein ligand, selection of the protein ligand. Then the drug designing cycle is going to be completed very rationally, and after searching of millions of compounds, we can analyze the structural activity relationship, and we can identify the drug likeness, and then we can work these selected prop molecules on cell lines, or then in silico, then in vitro, then in vivo. Then we can conclude that these molecules are very safe and these are very effective, and what are their inhibition constant and blah blah. So we can now using rational drug design, it is very promising, and the molecules, whether they are of natural origin or whether they are synthetic, they may easily go for the preclinical stages. What is the full-fledged strategy of molecular docking? We have some PDB files for ligand, protein and ligands. Then we have to prepare the protein in the form of some format which is known as PDB QT format. Then the protein PDB QT is generated. Then ligand protein PDB is converted into the ligand PDB QT format. Then the grid is going to be formed, and algorithm for proper algorithm is going to be selected for docking. And after this, the scoring of the topmost docked complex on the basis of energy, a score is going to be filtered, and the candidate complex or candidate ligand is going to be identified by using this simple strategies. So here we have to using the why we have to prepare the protein to energy and to minimize the energy, low pseudo energy based on the force field, because Autodock uses a specific force field which is known as umber, large surface burial, a small van der Waal overlaps, good hydrogen bonding, good charge complementarity, polar contact favored, and polar non-polar contact disfavored. These are the criteria for good docking orientations. Force field is one of the most vital part of docking and simulation. So it is a multi-parameter based entity. So here the force field, if you are using umber force field, there are then there is two state here for the ligand state and another for protein state, and then a combined protein ligand state. So here there is three. Here in the case of ligand, when the ligand is unbound, what is the potential energy? And then when ligands is going to be bound and unbound, then protein is going to be bound and unbound, and etc. So here ligand and protein of all energy is going to be calculated with reference to its entropy. Again, the total electrostatic hydrogen dispersion, repulsion, hydrogen bond, electrostatic and dissolution energy are going to be calculated. Here W shows represent weight constant, and the bracketed element is showing binding constant, and ET is showing the dihedral angle barriers. And the force field which is varying, which depends on the force, Coulombic force, which is dependent on the distance between the two atom. Here it is varies from van der Waal radii varies from 6 to the 12, while the hydrogen bond radii varies from 10 to 12, and dipole-dipole interaction varies from 1 upon R to the 3, etc. So all these parameters are going to be properly addressed while using the molecular docking. Okay. So here, how this whole process is going to be done and how pose is going to be predicted and how energy is minimized. Here, these things are going to be depicted here by the increasement of atomic distance, increasing atomic distance, energy energy decrease. There is a steep increase in the energy. So as the distances increases, there is a steep decrease in the energy. In the case of dispersion, in the case of dispersion energy, by decreasing distance, there is increase in the step. So here energy is going to be autodock force field is very vital for the calculation of energy parameters of the dock and ligand complex. There are several popular docking softwares in which Autodock is one of the most favorite and most preferred because it is free of cost for academic users. It is also having good citation. It is having some different facts. So Autodock is one of based on the Lamarckian genetic algorithms, and there are other dock, flex, fred, etc. So all these are having different algorithms on which these software are going to works. So Autodock, why we prefer Autodock? Because Autodock is having different features. We can prefer hydrated docking, we can do covalent docking, Autodock, zinc force field docking, rigid and flexible docking as such, blind and site-specific docking. All these different type of docking can be performed using Autodock. But there are some challenges in using Autodock, especially in the case of flexibility. If we want to flexible our protein, then it is very tough to, if we have a 200 amino acid protein, then each and every atom is going to be, it is very tough to flexible all these atomic coordinates. So flexible receptor docking is very challenging, and there is a requirement of high computation. Water is another challenge. Whether what should we do? Whether we should have removed all the water molecule from the protein available in the PDB, or we should retain some water, or we should eliminate all water. So all these things, selection of a water, whether the water is retained or whether the water is rejected from the protein ligand complex, protein ligand complex, it is important. So here it is also very and it is a deciding factor for energy of the computation. Another protomers and tautomers. Protomers and tautomers, most of the time, all that in the natural stage, most of the time, the ligands may be in the environmental condition, they may going to be charged. They may be having some charge. So in this case, if we are going to prefer the Autodock, we have to charge our own ligand, and charging of ligand and then docking, it is also a very tough job. So, and another is tautomers, large number of preparation of large number of tautomers. We have to generate a tautomer library of the protein before docking, and then we have to individually dock each and every protein tautomers to get the best possible docking pose. So these are some challenges in the protein docking. In the case of rigid and flexible docking, rigid docking can easily work on a standalone system, and we easily prefer. But for a flexible docking, high-end computation power is needed, high RAM and processors, etc. Rigid docking can be performed for large number of compound at a time, but flexible docking is not a choice. For large number of thousands of compound to be docked with protein, it is very tough task. And site-specific change in the binding pocket shape during binding. If you want to change the binding pocket during the binding, it is impossible for rigid docking, while it is a very easy flexible docking. So here we can partially make semi-flexible, we can make a complete flexible, and etc. So depending upon our own need, if we want having a small binding activity and we know that this is the exact binding activity, then we can make our site-specific docking make flexible. Otherwise, if we have no idea of whether what is the binding pocket and what exactly the size of the binding pocket, then rigid blind docking is the most preferable option. When we dock our protein, then the inhibition constant is going to be analyzed after energy calculation. So whether your ligand is how much it is giving your inhibition at which concentration. So there are three important amount can be represented in the amount of any ligand or inhibitor can be represented in millimolar, micromolar, or nanomolar concentration. So here the potentially active compound most of the time is having a inhibition constant of less than 100 nanomolar, while the moderately or weak active compound shows at 100 to 10 micromolar, while inactive compound is having Ki in the range of millimolar. So if we get a Ki value after docking in these range, we can easily identify that this compound is pharmacokinetically or pharmacodynamically will be effective or not. So Ki is one of the best parameter to decide or to select the inhibitor or reject the inhibitor after energy calculation. I am discussing what is algorithm, genetic algorithm, and why genetic algorithm is used in autodoc. So genetic algorithm is an algorithm which is based on the principle of genetics. Here each and every term of genetics is going to be involved. For example, gene, for example, number of population, for example, mutation. So all these terminologies are going to be used in the genetic algorithm. On the basis of these process, the autodoc calculates the whole set of probabilities whether the ligand perfectly binds or not. Here, autodoc is having some variants: autodoc vena, autodoc tools, and simply autodoc. And we can see that the citation of autodoc vena increases day by day, and it is about 11 million and more than this crossing. And this is an old statistic, but still you can say. So for running autodoc tool and following steps, you should have following step. I am just giving you a glimpse, the step in a single way, in a fast forward way, so that you can use by your own. So Windows 7 or higher version is necessary. Then you have to install Python 2.5 or higher version on your machine. Then autodoc tool can be downloaded from the given link. Then MGL tool can be downloaded from the given link. And then for batch docking, Perl is one of the most preferable language. So you have to install the Perl on your machine sequentially. Okay, so you have to download these things and install on your machine sequentially. What things are important here? When you have to download these things, you have all the icon will be generated on the desktop. You have to create a folder on the desktop in which you have to keep these files: ADT parameter batch file, ADT 4.1 bound file, then autodoc 4 executable file, then autogrid executable file, then vena executable file, and your target and ligand protein. So all, at least these 6 elements are necessary for a starting of your batch docking process. So here I have mentioned the link of the autodoc tool from where you can download, and you have to prepare. I am showing here, I have prepared an autodoc folder and I have kept all these things in this folder. So you have to do like this. Okay. And for the drug likeness evolution, I'm giving you what is the drug likeness and why drug likeness is important. So drug likeness means whether the drug is how much it is having efficiency of absorption, dissolution, metabolism, and toxicity. So all these things are going to be, especially for the ligand, we have to, there are so many databases or online servers from where you can identify the drug likeness of your molecule of interest. So you can prefer Swiss ADME, PKC, CSM, Admit Lab, Durolito of. So we can prefer all these online servers. So then the drug likeness of any organic compound is analyzed on the basis of Lipinski rules, Weber's rule, Bosch rule, Egan rule, Maggi rule, and all these. So at least these rules are going to be followed by your small organic compound. Otherwise, if there is a violation and serious violation in these rules, then after getting a good docking score and good Ki value, your ligand is not sufficient for proceeding in the clinical trials. So here, rules of oral drug, for example, in the case of Lewinsky rule of 5, which is one of the most central pivotal, which plays pivotal role for selection of your ligand. It says that the number of hydrogen bond donor should less than or equal to 5. Number of hydrogen bond acceptor should less than or equal to 10. For example, I have depicted here the donors, number of hydrogen bond donors, number of hydrogen bond acceptors, and the overall molecular weight of the ligand should be less than 500, and log p value should be less than. So all these having different more and less parameters with different numerical values. So if the if the ligand of your interest follows all these rules or having violation, less violation or not serious violation of any exceptional rule, then there is no problem. Otherwise, you have to seriously evaluate your ligand properly. So the step of docking is you have to download the protein from the PDB file. I have shown you the example. Or ligand, you can download it from Zinc PubChem database, or you can have, if you have to draw your ligand, you can use ChemDraw, Markvin, Sketch, etc. And you have to draw the ligand and you have to prepare the protein. And then you have already installed the MGL and Autodoc Vina. Then you have to follow. You have to prepare the ligand by using the step I have already depicted here. So you have to, in the case of protein preparation, ligand preparation, the most important factor is you have to, ligand preparation, you have to add the G-Steger charge. You have to identify the route. What is the route? Here I am showing why route is important. So here you have to identify a route which around which the whole protein, it is considered as the axis of the protein. So you have to identify the route, you have to detect the route, and then you have to give the number of torsional, how much torsions are available. So you have to give the torsions available in your ligand, and then you have to save the ligand in the .pdb qt file. So this is the Autodoc GUI, and you can easily perform the step by these simple ways. Okay, then you have to prepare the protein, and you have to prepare the protein by you following these steps. Here I have mentioned that if you have now protein in the .pdb file, then you have to, if I want to delete the total molecules, then no problem, I have deleted the water molecule, total water molecule. Or if we want to retain the water molecule, it is also okay, but the energy will vary in the case of hydrated docking or non-hydrated docking. So if we are preferring the hydrated docking, then retain the water. If you are preferring non-hydrated docking, then eliminate or delete the all water molecule. Then remove all heteroatoms. It means all other ligand which is already pre-docked complex in the form of PDB file. So most of the time, the protein database file is already have some ligands docked for preparation of the crystal structure of the protein. So you have to remove the heteroatoms from the PDB file. Then you have to check the missing atoms or missing residues, whether the protein is having some missing residue, then you have to identify the missing atoms and missing residue. If possible, the Autodoc tool itself will repair the missing atom. It will by probability using its genetic algorithm or other algorithms, it repairs the protein or it retains the missing amino acids. Then you have to add the hydrogen, and preferably we use the polar nature of hydrogen molecule going to be added. Then we have to charge the protein with Coleman charges, and then we have to disperse the total charge on the surface of the protein here, which I am showing in here. So you have to disperse the charge on across the protein, and then you have to save the protein in the .pdbqt format. So these two files are going to be prepared, and now these are the steps of Lamarckian genetic algorithm, on which there is how much population size we prefer. Most of the time, it by default it is having 150 number of population size. Then individual population we can increase. Basically, we should prefer up to the 300 for depending upon our own computation or our source of our own machine. Then number of energy evaluation, it is decided to be 25 lakh. So 2 lakh 50, 20 lakh 5 lakh. So we have to put these stage, or we can increase upon 25 to 50 lakh. And all other we have to we can crossover rate, we can increase it to up to the 8, 10, 12, etc. So we can prefer the by default setting, or we can increase depending upon our own size of the protein and size of ligand on our own resources. Similarly, there are 3 algorithms available in the Autodoc. Whether you can prefer the Lamarckian genetic algorithm, whether you can prefer the simulated annealing, and then or whether you can prefer local search. So most of the time we prefer Lamarckian genetic algorithm, and we have to optimize the parameter depending upon our own requirement. Then the preparation of grid, we have to follow how to prepare the grid. Now, when the protein is prepared, we have to decide the active site of what is the coordinate of active site in the protein. So in the case of site-specific docking, you have to select only the XY coordinate lying on the active site. But if the active site of the protein is not known and you are preferring blind docking, then you have to draw a full XYZ coordinate around the whole protein in the searching and the spacing of 5 angstrom, 0.5 angstrom to 1 angstrom. Not more than 1 angstrom is allowed in the Autodoc. So we have to allow in a search space of 1 angstrom maximum. So in this regard, we have to maximize our coordinate to get the whole protein covered for the blind docking. Then we have saved this file in the form of text format, and we have the center XYZ coordinate and size XYZ coordinate are depicting here. And then exhaustiveness of our analysis is 8, 10, 12, etc. Here now in this step, we have to install the Autodoc Vina, which is available at this website I am showing here. You can download the Autodoc Vina. For single ligand docking, just put all these things, receptor, ligand, and configuration file, which you have already prepared on a desktop in an Autodoc folder, and then follow this command vina.exe and configure.txt and log this file. Then it will identify, okay, your file are already present and the docking will be done accordingly. Okay, so all these operations are going to be performed from the command prompt. And in the case of batch docking, we have to write a Perl script in which the list of ligands are going to be prepared. And here the script says the Autodoc, this is our protein and these are list of our ligands. By sorting or selecting the protein using this script, it will identify that the whole ligand available on the text file is going to be called. Here in this case, we have to put all these files in the Perl bin. So in the case we are not going to run the script of Autodoc from the Autodoc folder or from the desktop. Rather, we have to do the whole execution in the Perl folder, bin folder. So we have to keep the protein PDB-QT file, protein ligand file, VINA executable file, VINA Perl script file, ligand text file, which I am showing here, and protein grid configuration file. All these files should be kept in the Perl bin. And here we have to use this command on the command prompt. Then it will ask name the file of the ligand. We will put the name of the file, and then after this it will identify the topmost ligands. Here these are, I am showing the Perl script for Windows and Perl script for Linux. By doing this exercise, now the terminal end of our analysis of docking is post-docking analysis, how we will analyze our result. So for analysis of the Autodoc result, we have to prefer PyMol, Discovery Studio, LeakPlot, Chimera, and Mastro. All these softwares are freely available and with some constraints or some limitation. So we can download all these softwares and we can prefer our academic research for their site as well as for their citation. So post-docking analysis and post-prediction is going to be performed from the LeakPlot, PyMol, and Discovery Studio. So this is the whole story. When the protein is going to be properly docked, then the image is going to like this. I am showing some image from my own analysis. We have identified how much hydrophobic contacts, electrostatic interaction, hydrogen-hydrogen bond interaction, pi-pi interactions, and all these things, how they are showing. So 2D plots and 3D plots, and all these things are going to be generated, and we can identify the this ligand is one of the best ligand that is going to be docked with the compound. So other resources, you can identify the resources from these websites and algorithms from this. This is the whole and soul story of molecular docking as such, what I prefer. So thank you for watching. Thank you.
Thank you so much, sir. So participants, you can ask your questions now. You can put your questions in the chat box, or you can unmute yourself. Please put your questions in the chat box, or you can unmute yourself if you're having any query.
Good evening, sir. Hello.
Yeah.
Yes, ma'am, you are audible.
Yeah. Yes.
Sir, my one question is, when we are doing batch docking,
Yeah.
Sir, in that, when we are analyzing the results, so
Yeah.
how we will split, because we will get the 10 docking confirmations we will get.
Yeah.
So when we are visualizing the result, for the visualization of result, how we will separate those interactions?
Okay. Yes. So this the best way is to use Discovery Studio, and you opening all these poses at a time, or you can open all these 10 poses in a different window, and then you have to identify the whole 2D analysis. How many pi-pi interaction are they showing? How many hydrogen bond interaction? How many van der Waals interaction? And whether there are steric clashes? All these things are very beautifully depicted by the Discovery Studio. So you can use the Discovery Studio, as well as all the poses can be opened in the LeakPlot analysis. You can identify the LeakPlot, or also in the PyMol, you can open them and identify in the 4 angstrom range or 5 angstrom range how many interaction, all different type of possible interaction they are showing. So these things can be visualized in these softwares.
Okay, sir, one more query. When we are doing Autodoc, not VINA, simple Autodoc, that time interactions are different, and when we are using VINA, some interactions are different. So how we will then justify?
There is no as such, in my opinion, there is no as such justification, but we can say that there is some algorithmic differences. Of obviously, Veena is having some different way of working, and Autodoc is having some other mechanism, because in the Autodoc Veena, these algorithms is not as such the parameterized by ourselves. The random, the by default parameters are going to be used by Autodoc Veena. While in the case of Autodoc, we customize our parameter very well.
Yes.
So I think this is the reason for the customization of our own parameter. This is the reason maybe for the differences in the results.
Sir, then for the results, means
Yeah.
it happens like when, as I did a few molecules which I have docked in Autodoc, it
Yeah.
shows the interaction with the catalytic site. But when I have done
Yeah.
with the batch docking with the Veena, so in that those interactions are missing. So
Yeah,
how I will means?
Yeah, you have, for example, if you, the grid size, you have changed the grid size. If there is
Yes.
difference in the grid size, by changing the grid size of your protein, the position will be changed. So always keep the grid size very safe. Always use equal grid size. What is using in Autodoc and what is using in Veena or other Pyrex or any software. The same grid, if you, I think if you use the same grid in each and every software, then there is no such so much heterogeneity. There may be similarity, of course.
Okay. Thank you.
Thank you.
Any more questions?
I think no more questions.
Thank you. Thank you.
So thank you, Dr. Sarwar, for giving such a insightful or in-depth elaboration of the step of the modeling, molecular modeling, docking, everything. The participants received this concept very well and hope they will benefit from this. So thank you very much again for coming and be a part of our FDP sponsored by AICTE. So this was the last session. So I request Roma ma'am to conclude the session or can end the session.
Yes, thank you so much, Dr. Beg, for such an interesting session. And I would request you to please share your PPT with us so that the participants will also get the information that was included in your PPT. And thank you so much for coming and accepting our invitation. And for the participants, we'll be meeting again tomorrow at 10 o'clock. We'll be having two sessions, 1 and 2. That will be followed by the exam for tomorrow. Thank you so much, everyone. Okay, I'll share the attendance link on the WhatsApp group. I'll be ending the session now. Thank you, everyone.